All right. Hello, everyone, and welcome to today's cryo-EM Current Practices webinar. You're joining the 3 national centers established by the NIH Common Fund Transformative High-Resolution Cryo-Electron Microscopy Program. My name is Christina Zamani. I'm a scientist and training liaison at the National Center for Cryo-EM Access and Training, NCCAT, and today I'm also your webinar moderator. I'm joined today by my colleague here at NCCAT, Ed Eng, and also Theo Humphreys and Lauren Hales-Beck from the Pacific Northwest Cryo-EM Center, PNCC, and Michael Schmid from the Stanford SLAC Cryo-EM Center, S2C2. S2C2 has invited today's speaker, Dr. Aashish Manglik, and Mike will introduce him properly after my colleagues from each of the centers give a brief overview and update on the cryo-EM resources we offer at no cost to the research community. For those of you new to our webinar series, cryo-EM Current Practices is an ongoing event that we host the last Thursday of every month at this same time. And we highlight particularly the methods that researchers are using to obtain and interpret the data they can collect at the national centers. So plug for next month's talk, James Chen and Brandon Malone from the Rockefeller University will be presenting on COVER All of the Bases, Structural Perspectives of SARS-CoV-2 RNA Synthesis. So you can save the date now for that talk. We are recording today's talk, and you'll find the recording along with those from past talks on the events page at cryo-EMcenters.org, along with the registration link for upcoming talks and other general information about the larger NIH Common Fund cryo-EM program. A couple of final logistics. We'll be using the Q&A feature for questions. So you can ask your questions there and also upvote other questions that you see. If you have questions directed to logistics or access at the centers, our panelists will respond to those directly in that Q&A box, and we'll save questions for Aashish for the end of his talk. And with that, I will turn over to the representatives from each of the centers, and Ed is up first. Take it away.
Hey. Hello, and I'm here at New York City where NCCAT is housed, and we offer dedicated instrument access for high-end instrumentation on one of our 4 Krios instruments, as well as a cross-training program. Right now, we have an upcoming call for proposals July 1st, but given the July 4th holiday, we'll accept some applications a little bit later. Something to note, right now, we are open for business, and especially on-site access. So if you want to spend a couple of days in New York for your Krios access, or a few weeks for your embedded cross-training, we welcome you here to the Big Apple. Hope to see you soon.
Thank you, Ed. Theo, you are up next.
Hello, everyone. My name is Theo Humphreys, and I work as a scientific point of contact for the Pacific Northwest Cryo-EM Center, or PNCC. At this time, we offer 1 proposal type for both single particle analysis and tomography, and that grants you up to 480 hours of microscope time a year for up to 2 years. At the PNCC, we have a monthly submission deadline for applications, and each approved proposal will be delegated a scientific point of contact, such as myself, to help answer scientific questions. The PNCC has 5 microscopes. We have an Arctic with a K3, a Krios with a Falcon 3 and a K3, 2 Krioses with a Falcon 3 and Bio Quantum K3, and 1 Krios with a Falcon 3 and Bio Continuum K3. And for sample preparation, we have a VitraBot like a GP2 and a VitraJet that's undergoing internal validation, but will be offered to users once that's complete. While we're still under some on-site restrictions, we're offering one-on-one remote trainings and small remote workshops. And we hope to have visitors on site soon again to offer workshops for sample preparation and microscope operation. We hope to work with you soon. Thanks.
Thanks, Theo. And Mike?
Hello, I'm Mike Schmid, and I'm one of the co-directors of the Stanford SLAC cryo-EM Center. As with the other 2 centers, we have access for high-end data collection and training. We have 3 Krios microscopes with K3 and Falcon 4 cameras. Some are equipped with energy filters. We have the normal selection of sample preparation equipment, and we are currently having some visitors on site for in-person training. We hope to have more people on site very soon for both on-site training and to complement our remote access for data collection with on-site access for data collection. Please come and see us. The website is there. Thank you. Now, it gives me great pleasure to introduce our speaker for today, Aashish Manglik. Aashish received his bachelor's degree at Washington University in chemistry and biology, and while there received the Spector Award. He got his PhD and MD at Stanford University, right here, in 2014 and 2016. And he was working with in the labs of Brian Kabilka and Georgios Skiniotis. He has received several awards for young and early investigator awards from Scientific American, from the National Institutes of Health, and from the International Narcotics Research Conference. His focus is on, as you'll see, cell signaling, especially the GPR system for cell signaling. There are thousands of GPCR receptors, most of which we don't know what they smell, and after they smell it, what they do. So, he's very involved in figuring out these orphan receptors for which we do not know their function. His talk today will be solving molecular puzzles in GPCR signaling with cryo-EM. Welcome, Aashish.
Great, thank you, Michael, for the introduction, and for giving me the chance to come talk here, and also for the attendees that have come. I must fully admit, at the beginning of this talk, that I personally am learning cryo-EM really through a remarkable collaboration with Yifan Cheng's group, as well as bolstered by both the S2C2 Stanford Cryo-EM Center and the Pacific Northwest Cryo-EM Center, as well as the UCSF Cryo-EM Center. So, my background really is in structural biology, trained as a crystallographer in Brian Kabilka's lab, and what I'll hopefully get across to you today is how cryo-EM has completely revolutionized our ability to peer into how GPCRs work. And I'll focus really on one system just to give you a sense for how quickly and how much more rapidly we're able to move compared to how where we were about 5 or 6 years ago. So again, the title of my talk is Solving Molecular Puzzles in GPCR Signaling. And really what I view modern structural biology is able to do, instead of being an end in itself where we get the first high resolution view after someone's entire PhD or postdoctoral experience, the rate at which we can get structures of GPCRs and signaling proteins is such that we can really start to fit in many puzzle pieces at once. And really, it's not just the structure itself that we get, but really gives us deep insight into the physiology or the biology of the system. So this talk will probably focus a lot more on the biology than the cryo-EM, mostly because that's really the perspective that my lab comes at it from. Okay, so what are we interested in? My lab is broadly interested in G protein coupled receptors. And this very huge family of proteins are incredibly diverse. And they sit on the cell surface and recognize a whole host of stimuli. So everything from hormones, proteins, ion, pH, light, or even force itself can activate one of these receptors. And the really neat thing about this broad family is that each one of these receptors, while they're slightly diverse, has a kind of a common mechanism of activation where these receptors then change shape and go on to engage a whole host of intracellular signaling proteins or signaling cascades like the cyclic AMP pathway, calcium pathways, or growth factor pathways like MAP kinase pathway. And perhaps what's even more remarkable is that mammals encode thousands of GPCRs. About half of them are just specialized for olfaction, as Michael alluded to. But the rest of these G protein coupled receptors control almost every aspect of physiology. So everything from neurotransmission, metabolism, feeding, you name it, is controlled by one of these receptors. And as a result, they've been perennially amazing drug targets for manipulating human physiology and restoring physiology in disease. So with this kind of importance in biology and in medicine, it's no wonder that there's a lot of effort to understand how these receptors function at the atomic level with the hopes that we can manipulate physiology more precisely. So what I'll focus on today is, not our work on many of these other physiological systems, but really focused on one specific signaling system that's deeply important in development and that goes awry in certain cancers, and that's the hedgehog signaling pathway. Just to give you a little bit of background on hedgehog signaling, this is one of these core developmental pathways that was discovered almost now 40 years ago by pioneering studies by Christian Nuslin-Volhard and Eric Weishaus, where they were looking at mutations that would disrupt normal development of Drosophila embryos. And this is the cover of Nature, from their pioneering studies, which showed that they could discover mutations that would disrupt this very normal segmentation of a developing Drosophila larvae. Again, here's a more recent figure. So you can see, for example, there's these very clear repeating units that are really important for determining certain axes for how the Drosophila embryo develops. And the hedgehog mutant was named because it basically completely disrupted this very specific phenotype, leading to something that resembled this cute little furry animal, or in subsequent years, the video game, which some of you may have played. Okay, so what does hedgehog signaling practically do? This is just in Drosophila. Hedgehog signaling is deeply important in guiding the body plans of essentially all animals. So for example, just to give you one example, if you're thinking about how do the cells in the developing, I have to do this on camera somehow, how the cells of the developing limb bud figure out which side is going to be the pinky or the thumb, it's actually a hedgehog gradient of this hormone that determines how that's going to happen. In the same way, hedgehog gradients determine what's going to end up becoming your head or your feet, what's going to end up becoming your front and your back. All of this is determined by hedgehog gradients in combination with a number of other morphogen gradients. And in adult tissues, when hedgehog signaling goes awry, people develop perhaps the most common cancer in all of North America, or certainly in Europe and in the United States, which is basal cell carcinoma, as well as a number of other cancers that are driven by aberrant signaling of this pathway. Now, how does this pathway work? And this is really where we operate, which is we want to understand at atomic level detail a number of the puzzles that have remained despite 4 decades or whatever of beautiful genetics and a beautiful biochemistry and cell biology to understand how this pathway works. So what we know at this point is that there's a number of components involved. We know that there's a protein called patched that has very high homology to transporter-like molecules that's putatively transports some sort of sterol, and depletes it from the inner side of the cellular membrane. And the constitutive activity of patched inhibits a GPCR-like molecule called SMOOTHENED, and this is what keeps the pathway off. Now, when the hedgehog morphogen comes along and binds patched, it's proposed that hedgehog inhibits whatever patched function is. This then releases the brakes on SMOOTHENED, and now SMOOTHENED turns on. And this activated SMOOTHENED inhibits the degradation of this transcription factor GLI, allowing it to build up and then turn on a whole host of transcription cascades that are responsible for hedgehog signaling. So again, this is, these parts are known, but really fundamentally what allows this communication to happen or how patch works remains very mysterious. And part of this mystery lies in the fact that there's a very intricate cell biological dance for all of these components. So for example, when the pathway is off, many of these components, for example, patch and another G protein coupled receptor, GPR161, exist in this very unique cellular membrane called the primary cilium. And when the pathway turns on, patch has been shown to be moved out of the primary cilium, SMOOTHENED moves into the primary cilium. This receptor GPR161 also leaves. So it's a very intricate dance that's happening. But at some level, all of this is controlled by very specific molecular interactions that have to happen. So a big goal that we have is to really understand, what are fundamental puzzles in hedgehog signaling. So for example, it's still not 100% clear how patch at the basal level inhibits the function of SMOOTHENED. Even the molecular function of patch, while it's been proposed that it's a transporter, we don't have incontrovertible evidence that that's truly the case. We still to this day don't really deeply understand or have a full understanding of how SMOOTHENED is activated and how it then triggers signaling cascades. And then finally, this orphan receptor GPR161, while it's known that it's a really important negative regulator of the pathway, we have very little understanding of what what manipulates its function to serve as a regulator. So each one of these questions, what we're hoping to do is to understand at a very deep mechanistic level. And in many ways, while we could go solve structures of many of these components, and some of these components, have already been structurally characterized, really our hope is not just to understand structures, but to build bridges between structure and function. And this is obviously a beautiful picture of the Golden Gate Bridge. And if you live in San Francisco like I do, perhaps you'll recognize that the Marin headlands are even more beautiful. And if you'd like to get over, again, the analogy here is that perhaps the function side lives here, and the structure is just plain old San Francisco. Okay, so if we think about this now from the perspective of structural biology, what we're able to do pretty often in structural biology, I say cryo-EM, while it's an incredibly enabling technique, is still limited by this, is that our view on structural biology is limited to low energy states of signaling systems. These are usually the stable states that are either crystallizable, or I would say, they're imageable. They're the ones that average together and that we can see in large numbers when we do a cryo-EM experiment. But really where biology happens, where signaling biology gets exciting and interesting, is are states that are actually responsible for transducing the signal, or for states that are responsible for transducing unique signals. And these states on average are unstable and dynamic. And as a result of resisted crystallography, certainly, and even for cryo-EM, unless you're going through really extensive methods of classification, it's going to be pretty rare that you can see one of these states. So our approach to this has been a little bit unique in that we think that instead of trying to go, collect enough images or collect enough crystal structures to go see one of these states eventually, perhaps we can find ways of actually stabilizing these states to actually image them or to crystallize them. And the idea here is one that we employed quite a bit when I was in Brian Kabilka's lab, which is that if you have a very dynamic protein, like for example, a G protein coupled receptor, perhaps you could identify an antibody fragment that recognizes that excited state of the protein, one of these active or intermediate states, and make it amenable to structural biology study. And this general idea, found a lot of traction for crystallographers, and we'd argue that it actually, again, enables us to build bridges between the structures that we can now solve much more rapidly by cryo-EM, but really what their functional relevance is. Now, just to give you a little primer on nanobodies, this is a structure of a conventional antibody. It's a pretty complicated molecule. It's composed of a heavy and a light chain, a heterodimeric protein. And it turns out that camelids like alpaca or llama, or camels themselves, make these really cool miniature antibodies that are just comprised of the single immunoglobulin domain of the heavy chain. But what's really cool about these miniature antibodies is that similar to conventional antibodies, that can recognize an almost infinite range of antigens. But on top of that, they're single little proteins that are ultra stable, and they're very similar to human antibodies. And for therapeutic purposes, they're very easy to make, and they're relatively cheap to make. Now, similar to conventional antibodies, which have about 6 very highly diverse loops that allow them to lock onto a bunch of different antigens, nanobodies have 3 loops. But what's really unique about nanobody loops is that they can inject themselves deep into the crevices of proteins that are either the active sites or the functional hearts of proteins. And as a result, they've served as really great ways of pushing and pulling on signaling systems. Now, in the past, most people have discovered nanobodies by actually immunizing these large alpaca or llama. And in the past couple of years, working with Andy Cruz's lab at Harvard, we've developed a number of completely in vitro methods to discover nanobodies. So the idea here is that we start with very large repertoires of camelid nanobodies just to understand how does nature build nanobodies. And what we've done over time is build very complex libraries, about 1 billion variants, billion unique nanobodies that we display on the surface of a yeast cell. So here, I'm showing 10 or whatever nanobodies. The unique thing is that in each one of these yeast is a plasmid that encodes for the nanobody that's on the surface. Again, this library is about 10 to the 9th, there are 1 billion unique nanobodies. And what we can do is go into this library, and for any given protein that we're interested in, we can do this kind of process. We can basically identify nanobodies that are specific to our protein by basically pulling on those yeasts that have a nanobody that binds to our protein, discarding all the rest, amplifying those yeasts, and then repeating the process. And for us, this takes on the order of about 2 days to run the cycle once. we run the cycle about 4 or 5 times, and thus are able to find nanobodies that are specific to any protein that we desire. And importantly, we made this technology available for free to basically every nonprofit group. And I think last time I checked, we've distributed our libraries to about 350 groups across the world. So with that, our hope is to, again, understand the structural biology of the hedgehog signaling pathway and you know, in many cases, many other GPCR signaling pathways. But really our lens into this, and what I'll describe today, is that we use these single domain antibodies or nanobodies to basically push and pull on these various systems to understand how they respond. And as a result, what we hope to do in the atomic level is understand how, understand the structural biology of one of these proteins, but then with functional studies, build bridges between a specific conformational state that we see and a specific function elicited by that conformation. Okay, so let me dive into the first example of our work. And that's really to understand the function of this protein called patch. Now, patch had been proposed almost 20 years ago to catalytically influence the function of SMOOTHENED. And pretty early on, there was a proposal by Phil Beachy's group at Stanford that patch perhaps sequestered a lipid modulator of SMOOTHENED. And then the general model that's been proposed in the field is that patch perhaps moves a sterol like cholesterol away from SMOOTHENED, and that's what keeps SMOOTHENED inactive. Now, over the past couple of years, the labs of Yan Ning, Shakun Li, and Vladimir Korokov, along with Phil Beachy and Yifan Cheng, have determined a number of structures of patch. And some of these structures are patched alone. Some of them are with this morphogen hedgehog bound. And what these structures have generally revealed, of course, is the general topology of patched, which is again, very consistent with the transporter-like molecule. And they've also revealed how hedgehog binds to patch. And the model now is that hedgehog binds to this extracellular region of patch, but then there's this N-terminal bit of hedgehog that dives deep into patch. And the proposal has been that this little long bit of hedgehog basically occludes the pathway whereby some sort of molecule that would be transiting patch needs to move. But that doesn't fundamentally answer the question of whether patch is a transporter. At a most basic level, some of the really pioneering work by Oleg Jardetsky and Peter Mitchell, almost now 60 or so odd years ago, suggests that for a transporter to be a transporter, fundamentally it has to complete a conformational cycle. So it has to, in a simple mode, basically have to go from an outward open to an inward open state, or basically go through a complete conformational cycle in order to move some sort of substrate. So we had a simple idea on how to basically approach this question of whether patch is a transporter. We wondered, could we develop a molecule that would trap patch in a single conformation? And there are two possible outcomes. One possible outcome is kind of like a G protein coupled receptor. We trap it in one conformation, and perhaps we trap it in an active or an inactive conformation that would lead to either pathway activation or inactivation. The other model is really consistent with the transporter, which is if we stabilize any conformation along a conformational cycle, that would inhibit the function of this protein, if it's a transporter, and thereby turn the pathway on, again, because of this double negative control of the pathway. Okay, so this is a project led by Yunxiao Zhang and Phil Beachy's lab. And what Yunxiao ended up doing is using our nanobody library to find nanobodies that would bind to patch in only a specific conformation. So what he did is he started again with our nanobody library, and he identified a mutant version of patch that was locked to be in a completely inactive state. So that was incapable of doing whatever its function, putative function is. And what he would do over multiple rounds of selection is identify nanobodies that were selective to this mutant form of patch over the wild type form of patch. I ended up with about 15 clones, but I'll tell you about one of them today. Now, really critical, one thing I forgot to mention is that really critical to this process is an experiment that goes like this, which is what Yunxiao and what we've done subsequently a lot in the lab is purify, for example, two different versions of the same protein. So in this case, what Yunxiao did is he could purify a version of patch that in this case was inactive, and that's in the green color, purify another version of patch, that's in this case the wild type protein in red. And then in order to find these very rare nanobodies that specifically bind to one conformation of patch, what he does is mixes these two proteins that are each labeled with a different fluorophore with a pool of yeast. And then he can do a simple flow cytometry experiment where on the y-axis would be all of the yeasts that are binding to conformation 2. On the x-axis are all of the yeasts that are binding to conformation 1. And then by simply drawing this red little gate and selecting out those clones, you can identify nanobodies that would be selective again to one conformation versus the other. So we did this process for patched again, and he identified a nanobody that binds to patched, binds only again to this mutant form of patched. And over multiple rounds of further improving its affinity, was able to get a molecule that not only binds to patched, but turns the pathway on. So, so this is data from some signaling data. This on the y-axis is just whether the pathway turns on, and the x-axis is concentration. And the best clone here is in blue here, which basically in a dose-dependent manner turns the pathway on to about 80% the activity of HEDGEHOG itself. We could also show kind of in an acute experiment that you could administer this kind of molecule in an animal and get pathway to get the pathway to turn on. So really bona fide, it's a bona fide patched inactivator and pathway agonist. Okay, so again, our hope is to build bridges between function and structure. And working with Yifan Cheng's group, again, one of my more early forays into cryo-EM, we were able to determine a cryo-EM structure of patched bound to this nanobody, which we call nanobody TI-23. And what you can hopefully see here is that this nanobody binds an extracellular domain on patched. And here again is the kind of ribbon diagram of it. Again, in these structures, similar to many other structures of patch that have previously been solved, we see a number of sterile-like densities, again, very strongly suggesting that patched is a sterile transporter. But here's where it gets really cool. When we compared where this nanobody binds to the binding site of HEDGEHOG, we find that their interfaces are completely overlapping. And that's what's being shown on the left here. So in blue, again, is HEDGEHOG, and yellow is this nanobody. And you can see that they both basically landed on the same interface. And I'll remind you that these selections that we did, we didn't use HEDGEHOG as part of those selections. So really what happened there is that this conformational selection that we do with our yeast display allowed the yeast or the yeast library to identify an allosteric hotspot on patch. Okay, so what has this taught us? Now, if we compare the structures of patched on its own that have been solved previously, in this case by the Beachy lab, or patched bound to this nanobody, what we saw is that this critical region, which we've termed the switch helix, has two different conformations, pose 1 for patch alone, and pose 2 when the nanobody binds. And now if we go back and look at all the structures of patch that have been previously solved, either bound to HEDGEHOG or not bound to HEDGEHOG, what we see is that this switch helix is in two different conformations. And in fact, this actually doesn't correlate with HEDGEHOG binding at all. So some of these structures in conformation A are bound to HEDGEHOG, others are not, and same for conformation B, some are HEDGEHOG bound or not. But so what we think is happening is that this nanobody stabilizes this switch helix in this specific conformation. And that this is associated with a constriction of this conduit in patch. That's right here. So again, in green here is a structure of patched, a conduit in patched without any nanobody, and in blue here is what happens when the nanobody binds. Again, this just reflects that switch helix moving, which basically then again creates a new cavity and a constriction. So again, even in the absence of that long little lipidic, tail of HEDGEHOG, this nanobody, by stabilizing a conformation of patch in this region, is able to inactivate patch and turn the pathway on, which again really suggests even more strongly now that patch functions primarily as a transporter, and simply by changing the conformational ensemble of patch, one's able to influence its activity and thereby influence pathway activity. Okay, so hopefully that gives you a little bit of vignette of how we're using single domain antibodies or nanobodies to probe transporter function. What I'll tell you about now is our efforts to understand how the pathway is turned on and understand this G protein coupled receptor called SMOOTHENED. So there's still fundamental questions, or some fundamental questions that remain in the field. One of which is, how does patch actually regulate the activity of SMOOTHENED, both at a distance, or what does patch actually remove or sequester from SMOOTHENED to keep it off? And then fundamentally, once SMOOTHENED turns on, how does that activated stimulus then get transduced inside the cell? So we had a goal to try to understand, early on, what is, how does SMOOTHENED actually turn on? What is the active conformation of SMOOTHENED? And while there's now a number of structures of G protein coupled receptors in the on and the off states, a couple of years ago, we had a very poor perspective on this SMOOTHENED family of proteins, or what we call family F GPCRs, and what their activation mechanism is. Now, one of the things that we've learned from a whole host of structural biology and biophysics, including a lot of spectroscopy work, is that G protein coupled receptors are incredibly dynamic proteins, both in the off state, but also in the on state. While they're bound to agonists, what we know now is that they're really quite conformationally complex. And if you think about this from kind of an energetics perspective, it's really the presence of a transducer molecule like a G protein or beta-arrestin that causes these receptors to become in one conformation. So in the absence of one of these transducers, these receptors explore many different conformations. And that's really what's made it very challenging to assess conformations of G protein coupled receptors. And I'd argue many other transmembrane signaling proteins by simply doing structural biology. I think really what you need to do to understand these excited states is to stabilize them in some way. Now, in order to really understand SMOOTHENED function, a couple of years ago, Ishan Deshpande in my lab did a very similar screen to what I described previously for patch. But here, the idea was to, again, start with one of our nanobody libraries and then progressively select for nanobodies that would only bind to activated SMOOTHENED. So here, again, it's a very similar scheme, but what Ishan did is looked for nanobodies that would bound, that would selectively recognize agonist occupied SMOOTHENED and would not bind inactive or antagonist occupied SMOOTHENED. So he identified a number of clones, which we've subsequently characterized. The first one, and this was when we were far more adept at crystallography than cryo-EM, we were able to get a structure of with X-ray crystallography. And this structure actually revealed quite a bit about how this pathway, how this receptor turns on. And it's published work, so I won't spend a ton of time on it. But here what I'm showing is a structure of SMOOTHENED, again, in an active conformation bound to one of these nanobodies, in this case, nanobody SMOOTHENED 8, which binds to the bottom of this receptor and again, props it open and keeps it on. What we learned from this structure, and that's one of the more remarkable findings from this study, is that while activated SMOOTHENED again binds to this synthetic agonist that we actually put in the mixture, it actually also comes down with a cholesterol molecule that's deep inside the heart of SMOOTHENED. So the field previous to this had primarily believed that cholesterol binding to this extracellular site was important for function. And again, solving a structure of the activated conformation of the excited state in this case revealed a cholesterol bound deep in the heart of SMOOTHENED that's actually, we think, probably more important for how the pathway works, and is really the cholesterol that patched probably sequesters away from SMOOTHENED. Now, at the same time, while we were focused on trying to get a crystal structure of an activated SMOOTHENED with one of these nanobodies, the labs of Cheng Zhang and Xiaokun Li have determined a structure of cryo-EM structure of SMOOTHENED, in this case bound to a heterotrimeric G protein of the GI class. For those of you who have been following the GPCR cryo-EM field, you'll recognize that there's been an absolute revolution in the structural biology of G protein coupled receptors by cryo-EM over the past 4 to 5 years. And that's really driven in part by the remarkable ability to very rapidly generate complexes of GPCRs with heterotrimeric G protein. So in some of our own work led by Antoine Cole, who was a graduate student at the time, and as well as when I was in the lab of Brian Kabilka and working with Yorgos Kiniotis, what you can hopefully see, from this early work is that, a 2D class average of a GPCR bound to a G protein is pretty darn easy to analyze. And you can see the receptor clearly there, different components of the G protein there, as well as an antibody fragment in this case that we discovered for this complex that's found a lot of use subsequently. That then, eventually gave rise to a pretty reasonable 3D reconstruction to enable structure determination. And really, this remarkable revolution in structural biology for GPCRs by cryo-EM is driven by the fact that most GPCRs, by their fundamental names, G protein coupled receptors, couple to G proteins, and if you can make this complex between a receptor and a G protein, this G protein essentially serves as a fiducial to drive alignment of the GPCR. So, what we've seen very dramatically is a proliferation of structures of GPCRs bound to their G proteins, and this is really exciting because it enables us to see receptors, bound to agonists or bound to their endogenous hormones. But one of the big challenges is that still remains is what if you want to determine structures of G protein coupled receptors and their off states? And really, if you want to understand how proteins function, you really need to understand multiple conformations in order to understand really what drives changes in conformation. So this has still been perplexing, and part of the reason why this has been challenging is because like many other very small membrane proteins, G protein coupled receptors have almost no extracellular or intracellular real estate that sticks out of the micelle or out of a nanodisk. And as a result, it's very hard to drive particle alignment, both from a size perspective, but also from the fact that these particles are essentially spherical. So we've been also pondering and working on using our single domain antibodies to essentially serve as fiducials for inactive state structures of G protein coupled receptors. And this is not a new idea. People have proposed this quite some time ago, and Yifan Chang, our main collaborator in all this work, has proposed that using antibody fragments or FAB fragments for small membrane proteins may function as far as a decade or so ago. But we wondered whether we could use again these nanobodies that were coming out of our screens to determine structures of G protein coupled receptors, with the hopes that perhaps again we could use structures of these nanobodies bound to their targets to build bridges between a conformation that we could observe and a function in a cellular context. So again, going back to this original screen that Ishan and my lab did, a different nanobody that he found from this screen we've worked up now. And this is a pretty curious nanobody. We call it nanobody SMOOTHENED 12. And what's really unique about this nanobody is that it's actually a partial agonist of the pathway. This is pretty rare. It turns out that finding antibody modulators or signaling modulators of G protein coupled receptors remains a pretty challenging feat. And what Ishan has found is that this nanobody can, on its own, very weakly turn the pathway on. So what you're seeing again on the y-axis is just whether the pathway is on or not, and on the x-axis is a dose response of this nanobody. We know that it's a partial agonist because if we fully turn the pathway on with HEDGEHOG and then dose in the nanobody, we see a decrease in signaling back down to this 25 or 20% level. So that's really very convincingly suggests that this nanobody, by doing whatever it does, clamps signaling at a very specific level in the pathway. So we wondered, how does this nanobody actually do that? And I'll be honest, when Ishan in my lab and working with Brian Faust, another graduate student, wanted to approach us by cryo-EM, I really didn't think this was going to work. And I'll also say that we'd spent quite a bit of time getting a structure of nanobody 12 bound to SMOOTHENED by X-ray crystallography with a maximal resolution that we were able to attain of about 3.8 angstroms. And well, that was probably good enough. We probably didn't need a better structure than that. And what's really remarkable is that, I think on their first try, Ishan and Brian working with, the S2C2 Center, were able to get a structure of SMOOTHENED bound to just this nanobody. So again, in green here is the 7 transmembrane domain of SMOOTHENED, which is a little bit of the extracellular region, and in orange here is this nanobody. Just to give you some kind of statistics, this is a pretty small membrane protein complex of only about 75 kilodaltons. I'd wager that the structured part is probably even smaller, probably approaching about 50 or 60 kilodaltons. And the overall, kind of nominal resolution of this structure is really quite good, about 2.8 angstrom resolution. So just a couple of other highlights. If we can get to the next slide. This structure was solved with with a pretty high affinity antagonist called SNAMP1, discovered again in Phil Beachy's lab about 15 or 20 years ago. And what's remarkable to me is that the structure with just this nanobody has a pretty good density for this molecule, so that's sufficient that it's completely unambiguous to resolve it. And for us, in other parts of our work in the lab, we're very interested in using structures to discover new small molecule modulators. And this is a very exciting space for us because in the past, it's been very challenging to crystallize GPCRs with low affinity molecules. And we're hopeful that the ability to get a cryo-EM structure of SMOOTHENED with this nanobody, relatively easily may enable us to now actually go discover new molecules and see how they bind, especially low affinity molecules that are pretty, early hits in a campaign. Now, the other exciting thing about this specific nanobody is that I told you this nanobody is a partial agonist, and in many ways it actually behaves as an inhibitor in a more endogenous signaling systems. And when we map the interface of this nanobody onto SMOOTHENED, which is again shown in these orange in this orange cartoon here against the surface of SMOOTHENED, which is colored by conservation, what we see is that parts of this nanobody engage pretty highly conserved regions of SMOOTHENED. And the reason why we think this is great and pretty exciting is that there's a lot of interest in developing SMOOTHENED inhibitors for a variety of cancers, but a subset of those patients that take SMOOTHENED inhibitors develop resistance to the inhibitors that have thus far been developed. So the exciting thing is that this nanobody targets a completely new allosteric site that hasn't been described to date. And we think that perhaps targeting this new site may be a great way of overcoming the resistance that's been observed in the clinic thus far. Okay, so I told you now about a couple of small mysteries that we've solved. We've worked a little bit on how patch actually functions. A lot of work that we've dedicated to understanding how SMOOTHENED is activated, both at the level of what are the conformational changes in SMOOTHENED that occur upon a pathway activation, and how can new poorly described allosteric sites impinge on pathway activity, with some exciting results perhaps for using very small membrane protein fiducials to drive particle alignment. The last thing I wanted to highlight, that's again all unpublished work, is our work to try to understand an important negative regulator of the pathway. This is a molecule called GPR161. And about 8 or so years ago, the lab of Peter Jackson at Stanford and really Psychet Mukhopadhyay discovered that this orphan receptor GPR161 is a really important negative regulator of the pathway. And the way that this works is that when the pathway is off, GPR161 constitutively turns on the GS signaling pathway to keep the pathway off. And then when HEDGEHOG signaling turns on, when the HEDGEHOG morphogen's present, GPR161 has to leave the primary cilium in order for the pathway to turn on. So while it's clear that GPR161 is a very important part of the pathway, it's been very unclear what actually controls GPR161 activity. And it's been proposed that GPR161 is a constitutively active GPCR. But more broadly, one of the things that's really unique about the GPCR, about all GPCRs, is that there's about 100 or so GPCRs for which we have absolutely no idea what their endogenous ligands are. So this is a nice, on the left here is a nice dendrogram from the GPCRDB that highlights, major classes, all major classes of G protein coupled receptors. And on the bottom left, this kind of big group here are all of the class A orphan receptors. Now, out of all of these orphan receptors, there's basically only one that's been structurally characterized to date. And that's one of the big challenges is, if you don't have a stimulus, if you don't know what the stimulus for a receptor is, it's very hard to go characterize it both at the cell biological level or the physiological levels, but also at the structural biology level. It's very hard to understand, how a receptor functions. So we've started to develop new biochemical approaches to purify orphan receptors and to start to work with them with the hopes that we can use biochemistry and perhaps even structural biology as a way of, a kind of a completely orthogonal way of getting into how orphan receptors function. And for GPR161, we've had a pretty exciting early success. Ishan Deshpande, again, in my lab, working with a really talented rotation student, Simone Harrison, another graduate student in my lab, Nick Copy, we're able to purify GPR161, again, bound to in this case, the GS, this is a type of GS heterotrimeric G protein, and one of these nanobodies that the Kabilka lab had developed many years ago to kind of lock everything together. And we're able to, again, working with the Stanford S2C2 Center, determine a pretty reasonably good cryo-EM structure of GPCR161, again, bound to its G protein. Now, the structure has revealed a few very interesting highlights. The first is if we look at the extracellular side of the receptor where a stimulus normally would be, what we note is that, let me get this thing to play well. Okay, there we go. So what we note is that what I've highlighted in red here is that the extracellular loop 2 of this receptor folds down on top of itself. And what we think is happening here is that this receptor has basically become self-activating, and there's a little bit of precedence for this kind of concept for another orphan receptor. In that, in order for this receptor to function, what it basically does is, in the absence of any kind of exogenous stimulus, this loop folds down on itself and basically keeps this receptor constitutively active. But on top of that, another really exciting thing that the structure has revealed is that we observe a mystery density that's consistent with the sterol kind of appended to the side of this receptor. And if we look at all of the amino acids that surround this sterol, they're highly conserved all the way down to echinoderms, which again suggests that, that we've potentially uncovered another sterol regulated receptor in this pathway. And again, this opens a new frame to try to understand how sterols regulate hedgehog signaling biology. So again, this project is at a very early stage, but we're hopeful that, you know, with having identified this this potentially new modulator, we might be able to really understand how GPCR161 controls hedgehog signaling. Okay, so with that, I'll kind of start to summarize. We think we're at a very unique time in structural biology, certainly for the GPCR field, in which our ability to gain structures, you know, far outpaces our ability to understand what they actually mean. And what this has led to is that structural biology for us has largely become not an end in itself, but really a means to see something for the first time and ask, you know, what is a relevant function of it? And certainly for orphan receptors, we're finding this more commonly that, you know, we make observations in new structures, and often that actually reveals new biology. A big push for us remains to try to build bridges between structural biology and function, both in the context of cellular biology, but also physiology. And for us, these antibody fragments are really a great way of doing that, for probing structure at the atomic level, but also interrogating what they do functionally to understand what the role of that conformation is in physiology or cell biology. So with that, I'll acknowledge again some of the key people that have enabled all of this work. Almost all of our cryo-EM work is done in deep collaboration with Yifan Cheng's group, has been deeply generous in almost every aspect, both training members of my group, you know, jointly advising a student, in this case, Brian Faust, who's again led a lot of the effort here. Almost all of the hedgehog biology work in my lab, in some ways led by a really talented postdoctoral fellow, Ishan Deshpande, and Nick Hoppy has been a really remarkable graduate student working at the interface of trying to understand orphan receptors and also using cryo-EM. We're a really remarkable lab of trainees that are you know, willing to work on pretty challenging projects. And you know, all of our hedgehog work is done in collaboration with Phil Beachy's lab at Stanford and Ben Myers, who's now at the University of Utah. Yunxiao Zhang's work I already talked about, and one person I didn't get a chance to talk about is Kaiwa Zhang, who's a postdoc fellow in Yifan Cheng's group and has done really beautiful work on hedgehog signaling as well. All of our nanobody work is done in collaboration with Andy Cruz's lab and his postdoctoral fellow Connor McMahon. You know, we're obviously very grateful for a variety of funding sources, but for this audience, what I'll give, you know, a couple of key shout outs to, you know, the UCSF cryo-EM facilities has been incredibly helpful, David Bulkley in particular, but really access to a number of cryo-EM centers, both the Stanford SLAC S2C2 Center, which is responsible for a lot of this work, as well as the PNCC Center, for which we've been able to get amazing data, but is on a completely different project, as well as the NCI cryo-EM facility. All of these national facilities have really enabled us to get data at a rate that really I couldn't have imagined a couple of years ago. So really want to thank all of these groups for making these resources so available, and for the training and just the expediency of being able to get collect data at these sites. So with that, I'll stop and take questions if there are any, and thank you for your time.
Thank you, Aashish. We have a question from Nam Nguyen. Can you please comment on why is it possible to determine the structure of smoothened in complex with the NB-SMO12 nanobody to such high resolution, but not a GPCR with an ICL3 fusion?
Yeah, great question. So for those of you who are perhaps not familiar with what an ICL3 fusion is, one of the strategies that had worked incredibly well for the crystallography era of determining GPCR structures is that instead of trying to develop antibody fragments to enable crystallography, what many groups did is, and this is really again pioneering work in the Kabilka lab, is to basically create a fusion protein into the intracellular loop of a GPCR. And really the primary thing that fusion led to, it was the ability to form crystal lattice contacts that then enabled us to get crystal structures. And the challenge with ICL3 fusions is that, there's basically 2 points of attachment. So getting that entire thing to be rigid, where the mass of the fusion protein is driving alignment of the GPCR, is really quite challenging. And in my lab, we've tried this quite a bit. It's worked to some extent. We've heard rumors where this has worked a little bit better for others. So it kind of, it's a very bespoke thing for each specific project. I think the reason why this nanobody approach works really well is because almost all of the mass is very rigidly coupled to a 3-dimensional epitope on a protein, and that's why we think that it basically drives particle alignment so well. And there's great examples of this, not just for GPCRs now, but for a number of other transporters, and certainly great examples of using full FAB fragments as well to drive particle alignment. So we think it's really a complex 3-dimensional epitope that enables this.
Thank you. I had a question about the comparison of the HEDGEHOG binding to the nanobody binding, and you say that the constriction is driven by the movement of a helix in patched itself, rather than any obstruction that's introduced by HEDGEHOG itself, like that long tail you originally pointed to as something that was kind of a candidate for that. Can you then look at what parts of HEDGEHOG are disposable, that you don't have to have that long tail, as long as you have something that moves that helix to that position of that in there? Does it give you more insight into what parts of HEDGEHOG really were important?
Yeah, so that's a great question. I think I probably misspoke or spoke too fast when I was talking about this. So what the field has done, Michael, is a version of that experiment. They basically chopped up HEDGEHOG and tried to figure out, what is the business end of HEDGEHOG? And what they've actually found is that this peptide that digs deep into HEDGEHOG is actually the most important part for inhibiting the function of patch. And in fact, this region that binds up here is important for affinity, but is actually dispensable for the activity of HEDGEHOG. So, that's why I think our study kind of flips that logic on its head. It's not just that lipid part, that blocking the conduit is important. You can actually block the function of patch, not just by plugging the conduit, but by sterically preventing its conformational transitions. So it's, HEDGEHOG itself actually probably doesn't necessarily limit this switch helix movement in the same way that the nanobody does, because what we see is that the nanobody really stabilizes one pose of the switch helix, whereas in the prior structures of patch that have been solved, some of these red conformations or blue conformations are both, when HEDGEHOG binds, the switch helix has been observed in both states. So clearly HEDGEHOG probably doesn't influence the conformation of this helix, but our nanobody, just by impinging on this helix, is able to recapitulate what HEDGEHOG does in a very different way, we think.
Thank you. We have another question. Is it possible to form trimeric complex with B2 ARGS without ligand in the purified system? If so, the GPR 161 being self-activating might not be unique? That's the question.
Yeah, that's a good question. So the biochemistry of whether you can form a trimeric complex and whether it's stable enough remains to be done. So I think I actually have done that specific experiment that Sung-Soo Yu is asking for at some point in graduate school. And I think that the answer is yes, you actually can form a complex even in the absence of an agonist. It's not a very stable complex, but it can be formed, and probably if one really desired to image it, probably could be imaged. We think that the GPR 161 self-activating feature is unique because we don't see this, if I could go back to that slide easily. I'll have to play the darn movie. Hopefully it sticks for a second. For most GPCRs, we don't see this kind of, this extracellular loop actually diving deep and occupying the same region that an agonist would occupy. So we think that, we think it's self-activating, not because we can form this complex, but because of the fact that this loop actually takes up the same space where normally an agonist would sit.
Thank you. So we have a question from Paula Flicker. You are using nanodiscs for the EM in these cases, correct?
Paula, that's a great question. And I'm slightly embarrassed to admit that most of this work is actually with detergents. So we're in the GPCR field, unfortunately, has been a little bit slow to adopt nanodiscs. And that's partly because, in the past, when I was in Brian Kabilka's lab, this is again, work really led by Antoine Cole, working with Yorgos Skiniotis. We tried for a very long time finding an ideal detergent, an ideal setup to do cryo-EM for GPCRs. And we landed on was a very specific mixture of two detergents that's really, and it's been incredibly enabling for the entire field to get structures of GPCR, G protein complexes. So mostly for reasons of expediency, we haven't gone back and done really careful work with nanodiscs. My lab is slowly moving, to using nanodiscs. There's a couple of great examples out of Gerhard Wagner's lab, as well as working with Alan Brown, as well as Dan Rosenbaum's lab of structures of GPCRs now in nanodiscs. But by and large, that still hasn't caught on in the field as kind of the de facto mode of getting these structures. But great question, and something that we're striving towards.
If I can ask a quick question. So you maybe touched on this, going back to your nanobody 12 smoothened structure, you said the connection was not very flexible. So I was wondering if you could speak to sort of local resolution, given you're using that nanobody as a fiducial. Does it look good in that particular structure? And then given you've now looked at a couple of things, are there any trends in terms of local resolution when you're using these nanobodies as fiducials?
Yeah, so I will say that this is the only example that we've gotten. This has emboldened us to do more nanobody-driven fiducial. I really, and this work is very hot off the press. I think we only got the structure about 3 or 4 weeks, or maybe 6 weeks ago. We are now emboldened to try other nanobodies that we've discovered over many years and to see if they're sufficient to drive particle alignment of GPCRs. My gut feeling is that because the center of mass of this nanobody is not that far away from kind of the center of mass of GPCR, that we won't get these very profound local resolution problems. For the G protein complexes, this has actually been a problem for the field, where a lot of the center of mass of the G protein is, drives particle alignment for the G protein, and you get these beautiful high resolution views of the G protein, which are basically the same for every GPCR complex. And then the part that you care a lot about, which is the drug binding pocket, has just abysmal resolution, and you just can't even see what we care about the most. So we think that this nanobody approach certainly will hopefully have less of that. But then again, we don't know how successful this will be over many different receptors and many different nanobodies. This might just be an edge case, or it could be more common.
Well, we're at time, actually. So if there are any burning questions, people can type them in, but most people probably have somewhere to run off to. So thank you very much, Aashish. That was really gorgeous maps that we saw there. And we hope everyone joins us again. at another one of these seminars. And, from the cryo-EM centers, please let us know how we can help you. And hopefully we will see you again.
Thank you, Aashish.
Thanks a lot, everyone.
Mhm.